Hello, this is Isaiah and Nick, and today we're going to be talking about cystic fibrosis and an alternative solution to the problem by using CRISPR. Just before we get started, we just wanted to kind of establish the idea of what a genetic disorder is. And so these type of disorders are usually conditions caused by mutations in one or more genes. And the issue that kind of is going on is the fact that the gene mutation usually affects a protein, and as we've learned throughout lecture and both lab, proteins are absolutely critical to life. And so with that, by disrupting a very small piece to the bigger puzzle, causes pretty much drastic effects. The motivation behind our project arose from trying to find a way in which we could integrate concepts and techniques learned throughout bio lecture and lab. We found that in looking at a real-world issue such as cystic fibrosis, concepts such as mutations, DNA, and genetics go hand in hand with the research that goes into it. Throughout our exploration, we took notice in the vast usage of PCR, gel electrophoresis, and sequencing in the research involving the usage of nucleases for the disease. Overall, we were excited to share in hopes of all of you gaining a further understanding as to how small changes at the molecular level can drastically affect the human body.
Cystic fibrosis is most common among people of Northern European ancestry and affects about 1 out of every 3,000 newborns. About 1 in 25 people are carriers. It was first recognized as a specific disease by Dorothy Anderson in 1938, with descriptions that fit the condition occurring at least as far back as 1595. The name cystic fibrosis refers to the characteristic fibrosis in cysts that form within the pancreas.
Now, what we learned and hope for you guys to understand is that with cystic fibrosis, this involves a deletion mutation. And a deletion mutation essentially changes the number of DNA bases by removing a piece of DNA. In this case, it's 3 letters of the DNA. But there are other mutations that are out there which we learned about. Some of these include a missense mutation, a nonsense mutation, an insertion, a duplication, a frame shift, and a repeat. Just by looking at these and learning about them, we were able to kind of get a better idea of how to differentiate between the mutations just by their definitions.
It is also an autosomal recessive disease that requires both parents to be carriers of the abnormal gene in order for the trait to develop. This is usually a mutation on one of the first 22 non-sex chromosomes. Some other examples of autosomal recessive diseases are sickle cell anemia and Tay-Sachs disease. Mutations that cause cystic fibrosis occur on the 7th chromosome. The transmembrane conductance regulator gene causes the CFTR protein to become dysfunctional. Chloride ions are not able to help move the mucus and bacteria across the lungs' surface cells. This causes the mucus to become thick and sticky and is not easily moved across the membrane of the lungs. Our body normally secretes a chloride ion in the form of salt to help absorb the mucus and keep the bacteria from building up. However, because of a mutation in the CFTR gene, the membrane is not able to release the salt, and this causes the mucus to build up and allow for bacteria to sit on the membrane of the lungs, causing infection and the sensation of drowning from the fluid buildup. Some symptoms of cystic fibrosis are salty tasting skin, frequent lung infections, and poor growth or weight gain for young children. Some treatments are wearing a vibrating vest that breaks up the mucus and allows for the normal flow of mucus throughout the body and especially the lungs. There are better treatments and possibly cures in the near future, however. The Cystic Fibrosis Foundation estimates that with the advancement of gene therapy, the mutation can be corrected and cystic fibrosis will be eradicated. Cystic fibrosis is a genetic disease, and for the child to have the mutation, each parent must have one of the cystic fibrosis defective genes. However, there is not a 100% chance that the child will develop the disease even if both parents have the defective gene. The picture to the right is from the Cystic Fibrosis Foundation and shows how it can be passed down from the parents to the child. There is a 25% or 1 in 4 chance the child will have cystic fibrosis when both parents are carriers. There is a 50% or 1 in 2 chance the child will be a carrier but will not have cystic fibrosis. There is a 25% or 1 in 4 chance the child will not be a carrier or will not have cystic fibrosis if both parents are carriers.
The image on the left serves as a visual representation of what Nick described in the previous slide. As you can see, on the top of the airway epithelial cell located in the lung, a CFTR protein serves as an ion channel for chloride ions to pass. You may also see that in a normal cell, the mucus flows in one direction. This is due to the power and the recovery stroke of the hair-like cilia that sit on the exposed portion of the cell. We will explain a more in-depth look at the mutation a little later, but you can see a dysfunctional or non-existent CFTR protein will result in the thick packing of mucus that smashes the cilia. With a thicker mucosal membrane present, there is an increased concentration of bacteria and the horrid sensation of drowning. We started our research looking with general reliable sources such as mayoclinic.org and the Cystic Fibrosis Foundation. We found treatments for the disease, of course, include medicinal therapy, but these only help ease the symptoms. So we asked, why not attack this problem at the molecular level? This brought me and Nick to begin to look into primary and secondary sources that showed how research involving CRISPR proves to have a very bright future for fixing the problem at its core.
The most common mutation, called delta F508, which accounts for 70% of cystic fibrosis cases, is a deletion of one amino acid at position 508 in the cystic fibrosis transmembrane regulator protein, which includes the last cytosine of isoleucine 507 and 2-thymidine of phenylalanine 508 codons. Thymidine is critical for syncing G1 and S phases of interphase. You can see how just deleting these 3 out of over 1 million different nucleotides causes the entire body to not be able to process the mucus and bacteria correctly.
So in order to get a better picture and a further understanding of cystic fibrosis and kind of what goes into it, one of our secondary sources kind of explained therapeutic strategies for cystic fibrosis and how CRISPR can kind of be integrated into it to solve the issue right down at the molecular level. This diagram shown right here kind of breaks down the different mutations, and we kind of went into the class 2 mutation, and from there we looked at how CRISPR could be used to pretty much solve that issue. As you can see, cystic fibrosis consists of multiple different mutations, but the one that we focused on was the F508 deletion, which results in the deletion of phenylalanine, which is a hydrophobic amino acid. And the reason why we focus on this is because there are drugs that look to attack the symptoms that occur with the deletion, and we can go a little bit further in looking at the molecular level and what goes on there, what goes on around there, as well as this is about 70% of the mutations that are caused within the disease. So looking at a little bit more severe disease, it's a little bit more prominent, seemed more relevant for us to look into. So this secondary source kind of explained how nucleases, which are enzymes that pretty much cleave the chains of nucleotides and nucleic acids into smaller units, we'll see this kind of what you'll see in DNA replication. It's a very critical process that has to happen for DNA to then replicate. But we're pretty much looking at how CRISPR works as molecular scissors to essentially go in and open up that site, cleave it, and actually try to correct the mutation in that sense. Now, it is critical to understand there have been other attempts in looking at gene editing, especially for cystic fibrosis, using other nucleases. Some of these include zinc-fingered nucleases. There's also another one called TALEN, which is transcription activator-like effector nucleus. But CRISPR is probably the most promising as to the sources that we were looking at. There's another primary source that we looked at, which kind of evaluated the different nucleus options, and CRISPR pretty much was the chosen one.
In all simplicity, CRISPR is like a giant pair of molecular scissors. There are 3 basic components of how CRISPR works. The first component is the gRNA, or guiding RNA, which assists in finding the target site on the DNA strand. The second component is once the site is found, the nuclease will begin to pull apart and detach the DNA. And the third component is when the detached DNA sequence will be replaced by the template DNA.
So here we have a primary source that we use that kind of went over using CRISPR for the CFTR gene for functional repair inside an intestinal stem cell. This was these cells that were obtained from cystic fibrosis patients. And what you can see that the enzyme was actually pretty effective in correcting the mutation. And on the right, you can see how that's the F508 deletion we were talking about. So overall, CRISPR does have a bright future. The biggest issue that we've seen in a lot of these primary sources that utilize CRISPR was trying to find a way to actually get CRISPR to be implemented or introduced into the cell. Obviously, with experimentation, you have a little bit more control, but the bigger picture is looking at as to how to integrate this into a real time or just real like situation.
For example, a quote from our primary source states that they have isolated and expanded adult intestinal stem cells from 2 cystic fibrosis patients, corrected the mutant F508 allele using CRISPR-Cas9 mediated homologous recombination, and demonstrated functionality of the corrected allele in the organoid system.
Now, just for an overall summary of what we kind of understood from our small little research process was that, so we have a deletion at the F508 location on the gene. This involves a deletion of phenylalanine, and it's a 3 base pair deletion, as you can see in that photo right there. This particular deletion makes up the largest percentage of cystic fibrosis patients. One of the biggest advantages with this particular issue is that it's monogenetic and that it occurs just in one gene. That advantage is easy to go ahead and target rather than having to target multiple different variables. Again, one of the earliest stage troubles with using gene editing is the fact that they have to isolate the cell outside of the organism rather than actually being within the organism. So again, there's still a lot to go. There is a big future with CRISPR. Other questions could be, are there even other possible ways to correct these type of mutations? Another one we saw that we kind of glanced over was the process of base editing, which is kind of a direct swap in point mutations, but it didn't seem to correlate quite with what we wanted in our project. But going forward, it would be another cool topic to kind of look into. So overall, we really enjoyed the experience, and we thought that we were able to find ways in which to incorporate ways that we incorporate concepts that we learned within class and actually apply what we know, and to try to understand a little bit more difficult text that stretched farther from the class. But just by using what we knew, we felt we learned quite a bit.